I'm going to talk about clinical trials in the UK, how we got to where we are today, because that was my original remit. And that also gives us a great opportunity to discuss the FLAIR study and where we might follow on from that, because I think that fits very nicely with what Helen told us about IMV. And then the final bit is going to be a little bit about relapsed CLL. We haven't actually discussed all of the standard relapse therapy, and so I've got some slides from Marano actually, but really that's as a lead on to what we might do beyond that. So Pete Hillman gave me these slides. He gave a fantastic talk of looking back at clinical trials in the UK as a memorial lecture for Professor Kotowski, who sadly died last year. And so I just sort of do a bit of history, and Pete put these really nicely together, and it was a really nice memorial lecture. So just a couple of minutes on that. Daniel was a man who came to the UK in 1967 from Argentina, and he actually set up the first MRC leukemia unit and became very influential in a number of diseases, but this talk is about CLL, at both the Hammersmith and then later on at the Royal Marsden and the Institute of Cancer Research. And, you know, this is a man who's cited, who published 800 papers in peer-reviewed journals, was very influential in the CLL scene in the UK, and then making the UK a dominant force in the international scene, for instance, through working through the international workshop in CLL. And of course, we got that meeting in October. So he pioneered trials with the CLL, and these were originally the MRC, and it was amazing hearing Pete describing it all, actually, because all of the things we've been talking about today and everything else, I mean, there wasn't even a clear diagnostic criteria for CLL and that, and then the Ryan Binet staging appeared. And of course, what they were using was pretty simple stuff. I mean, with oral chlorambucil. So this trial, the CLL4 study, was an interesting concept because everyone in the UK came together and adopted that. And of course, now I'm in the chair position of the NCRI. That is the forum that we are currently using to discuss that, and that will continue despite what anyone may have seen coming out. We will continue as a strong UK force taking CLL trials forward. There was the concept of central pathological analysis and biobanking, and I'm just going to mention about that, and also a great publication area. And just so everyone knows what the CLL4 study was, this was looking at different chemotherapy regimes and using chlorambucil, certainly no antibodies around, or single agent fludarabine, or the combination of fludarabine and cyclophosphamide. And those are the survival curves from the Lancet publication in 2007. And this is what showed that combination fludarabine and cyclophosphamide in terms of progression-free survival, and of course that's what we really talk about today. All those curves we've been seeing this morning are PFS curves, because as we know, overall survival, difficult one to demonstrate today, because really I think we're talking about such brilliant treatments keeping patients alive for so much longer. But of course, also that in fact, until FCR came along, there was no treatment that was shown to be better than chlorambucil alone in terms of overall survival. But this taught people how to use chlorambucil well in its in the regime that it was used, which was not on and off all the time, but the monthly pulsing. The showed that FC was a superior regime, and that's where the FCR regime grew out from. Also, and a concept that came up in talks that we had recently at EHA, that you need to be following patients up over a very, very long time to really understand what the significance of your treatment is over for the CLL patients. And this is a disease where we're talking about decades of following patients to really learn. And the CLL12 study actually we saw today from Roz just illustrating that point, but the amount of follow-up that we go there. Also that biobanking was important. So these were the NCRI phase 2 studies that followed on there, and it's interesting that an awful lot of them have got Campaf or alemtuzumab in there. So that just shows that was the thought for these difficult to treat patients, what you do with them, and really in a series of another trials from 1998 to 2015. The phase 2 studies on ALPMAR and ALTA, which have again been very good for learning about CLL biology. And then these other studies here, Galactic was a study in consolidation, Rialto was a study for the less fit patients. And something that came out of the whole trial program was having a dedicated UK CLL trial biobank, which is still alive and well in Liverpool. And that means that researchers can access trial annotated banked samples in order to understand more about the biology of a disease. And in fact, we had a meeting relatively recently where we were trying to bring the all researchers in CLL, both clinical and non-clinical and clinical academics together, and just think a little bit about what expertise we have in the UK. And this is complicated diagram, and I'm not really, but just to say what we're now thinking that we should be biologically asking about disease as categories there, and also the number of centres in the UK. And it's always difficult putting these kind of lists together because I'm sure there's going to be some centre in there which thinks it's very active in CLL research, which isn't on that list, and that isn't due to design. That's due to the fact that we do have a lot of fantastic centres researching CLL and often using clinical trials as the platform to do that. So it seems a good opportunity this morning to talk about our current major studies, of which FLAIR is one, and quite how we got there and where we're thinking of going beyond. It's always difficult to give talks about beyond because there obviously things get discussed a lot. But one concept that came out of the whole NCRI troop is to use this idea that we could do smaller phase 1, phase 2 studies, which would have biological correlative studies, but that actually would inform our large phase 3 trial, in this case being the FLAIR study, which could then be adapted by the kind of things that we were describing there. And one thing that struck me that when Pete gave the talk in the BSH was he said it took him 11 years to negotiate the FLAIR study, and I just thought, wow, it just shows how difficult things can be. So the TAP trial program that we have done recently, and people on this call will have been involved, was the Icicle studies and the phase 2 Clarity study, which I think really paved the way for this whole concept of ibrutinib and the venetoclax together, and thinking about delivering that in a MRD or a biologically guided fashion. And that information from Clarity is then informed quite what we've done in FLAIR. So they were helpful in doing that and explore the concept of deep remissions using MRD as a primary endpoint. Now, the problem with using MRD as an endpoint is it's not recognized by the regulatory authorities. So I think many of us would like to use MRD, despite all the caveats we had in the last session, as an endpoint in CLL trials. But if the regulators are not going to actually accept it as an endpoint, then it's very difficult to see how those regimes will then get licensed. And I guess that's the real problem we have with the current doublets, is that the academic community is doing some different things to the more pharma-sponsored licensing studies, but we end up with a license regime. So Icicle, those of us, this was a wonderful way of accessing ibrutinib when it was first available as a drug, but not licensed and certainly not funded. And it had two cohorts. It had a treatment naive group who just received ibrutinib monotherapy on their own, and a relapsed refractory where obinutuzumab was given in addition to that. And just really to show this slide that just you could monitor MRD over time, and this is all done by flow cytometry in the labs at Leeds, but also the much deeper remissions one gets in terms of MRD with a combination of obinutuzumab. Now, that may all seem really fairly obvious now, but I think when these studies were done, this is what we were learning. Clarity has been published in 3 years ago, but it's also continues to be updated. It has just now officially cut, but this is the trial where we were able to deliver combination ibrutinib and venetoclax to those relapsed refractory patients, and we did it in an MRD tailored way in terms of we were doing a lot of bone marrows and scans in patients, and so all the comments that George asked about scans and marrows at the beginning. Obviously, in this study, you had a lot, but that was telling us about the disease, and I don't know quite why that happened. These were the most recent updated PFS and overall survival curves for this really difficult to treat population, some of them. And they're very impressive. And this is giving IMV in an MRD directed way, i.e. here you're getting, up to 3 years of venetoclax and continuous ibrutinib. And we have some patients on the Clarity study at King's who were refractory to multiple treatments and are now actually completely off treatment because they've obtained MRD negativity and doing really well. And it's just an amazingly powerful regime given in this way. And interestingly, Tal presented this data at ASH last year, which showed that it was the patients who really quickly became MRD negative who are the ones who've benefited most from these regimes, and describing two different groups of patients. One where you get a very rapid depletion in MRD versus and that translates into better CR and remission rates compared to those who take longer and presumably are the ones that we need to give longer treatment to. So here I am. So FLAIR started off as this concept of using a chemoimmunotherapy regime, FCR, so the FCR eligible group, compared to ibrutinib and rituximab. And the point being here that if we look at this, that we originally have the Icicle study and then the Icicle amendment, which was the study which allowed the addition of obinutuzumab, and then Clarity. And what we were able to do was to then adapt the design of FLAIR to actually start asking different questions. So a trial that started off looking at a comparator of chemoimmunotherapy to then we were giving ibrutinib and rituximab, although in fact it was possible to amend this because it became clear that the addition of rituximab is probably unnecessary in CLL in the frontline study. That was the Alliance study that showed that, so we were able to amend that. But then bringing in this ibrutinib and venetoclax regime, and we've been able to continue to use the arms that you've got in it. So you've now got these 3 arms in the study: FCR, ibrutinib, and venetoclax. And what we want to do is to have these readouts. So we've read out already the FCR ibrutinib in an initial reading, and that was presented at ASH last year. And now we've got data which we're presenting on the ibrutinib versus ibrutinib venetoclax combination, and that was initially presented at EHA last year and at ASH as well. And I'm fairly sure we'll have a further update at this ASH. And all these other things that we can compare. So and that's showing this amazing adaptive trial design. I will highlight this again later, but we've got a continued phase 2 study, which is for this high-risk arm, and we can debate that because I think that's an important question. I know there are concerns about using ibrutinib perhaps in the frontline setting for this group, but I think we should discuss that. So I'm not going to talk about FCR versus IR other than just show this progression-free survival curve, and FLAIR confirmed that in terms of progression-free survival, that FCR was an inferior regime, and ibrutinib rituximab was a winner. Now, that's, of course, we'd also seen in the ECOG study, but I may say that FLAIR has a very, very high number of patients on a one-to-one randomization, and actually allows us to look at biological subgroups in a way that I think other studies won't do. And George was the one who said that the GLOW study actually had very few patients in it in order to be able to really be able to dissect out different groups. So we can look at different biological groups in the FLAIR study for this data. So FLAIR actually uses MRD as an endpoint, and the concept is that once you've obtained MRD negativity in terms of a study that is defined as less than times 10 to the 4 in the bone marrow, initially examined in the blood, that you actually just carry on giving treatment for the same period again, and that potentially could be maybe a cure for CLL. And therefore, if a patient obtains MRD negativity at 12 months, they will then get 24 months of treatment. But if it takes them 18 months to become MRD negative, it's then 36 months. And this is how we do that. We do a lot of peripheral blood MRD testing. So patients have been coming up for the last 6-7 years, getting MRD taken every 6 months. For a standard of care trial, that is not standard of care practice. We did a bone marrow at 9 months because that is the traditional time point in chemotherapy studies to assess for response. So that's really why you get that 9-month bone marrow. And I must say that all the patients I've had, let's say just on ibrutinib, get a bone marrow there, and I say, well, you've achieved a partial remission. That's not really very meaningful for the patient, because I say to them, I've done this because you're being compared to the chemo arm, but in fact, I'm extremely pleased with your response. And I think there's a bit of language around there and what we use in trials and how we explain to patients, which has been interesting learning. And the concept is that you hopefully can stop treatment. So of course, you need to become MRD negative. And if you're just on ibrutinib monotherapy, that's very rare. But also you've got the ability to be able to restart treatment if the disease is to reappear. And that, of course, is restarting for MRD positivity. It's not restarting for progression. So that's an important thing for us to remember. And these are the original readout from last year, which was the primary endpoint for the I versus IV was what is the MRD negativity state of patients? And what we see is that ibrutinib doesn't achieve that at all, but IMV really impressive rates of MRD negativity, and moreover, actually quite similar between peripheral blood and bone marrow. And I must admit, in clinical practice, I would not really do an MRD for a bone marrow for MRD status, because there's not a lot of data out there suggesting that you can get it from the blood if you've not got recent exposure to antibody. And just to finish off this section, what did we also learn, and what are we also learning, and we're going to learn more, is that you become MRD negative in the blood quicker than you do in the bone marrow. And these are the curves showing that. So I think there's still going to be masses of data to learn from this, and it's going to inform future trials and what we do. But of course, as we've been discussing in the license setting, we're only giving IMV for the 15-month fixed therapy. And just as a reminder to everybody, there is still a high-risk arm of FLAIR open, and that is examining P53 disrupted patients, whether they may benefit from an MRD-directed ibrutinib venetoclax therapy, or indeed for ibrutinib monotherapy. And I personally feel that if you've got a patient who has got nasty genomic disease as defined by TP53 disruption, and it's acceptable to give ibrutinib, i.e. they are not got a significant cardiac history, I think this is still an excellent study for patients who are P53 disrupted. Finally, on this section, static is now coming on board, and this is asking the question, can we stop ibrutinib monotherapy? Because patients on FLAIR actually get 6 years of ibrutinib, and A few of those patients have fallen off the back end of that because static's not yet open everywhere. But in fact, I think we're increasingly realizing that maybe absolutely continuous BTKI therapy until intolerance or progression may not be the right way to do it. And the concept behind static is to answer that question. And at the moment, it's really mainly for patients on the flare, or let's say the Clarity study, but I think it will expand more to other BTKIs. And actually, I think if we can adopt this and amend it appropriately, and that again is using that risk adapted sort of design, I think we could get some fantastic information out of static. I'm going to change to tact now. I'm going to mention a little bit more about what studies we're thinking of later, but I just now wanted to go to relapsed CLL, and I am very aware of the time, so I will stop at 10 past. When Helen and Roz and I were exchanging emails over the last few days about what we were going to talk about today, we actually realized that we hadn't really entirely covered the relapsed area of CLL. We've learned about using relapsed therapy in terms of acalabrutinib and zanubrutinib, and indeed we haven't really covered it today, but there is there's lots of data out there using ibrutinib in the relapsed setting. But we hadn't really thought about everything, and I don't actually think we've discussed the Murano study at all, which we're all just, when it was first presented, this was such an exciting time, for the CLL being able to use a treatment at all, and that was being updated. So we now got 7 years of follow-up data on Murano, and that was updated in the same session that CLL12 was presented at EHA, and actually just showing the importance of long-term follow-up studies. So to remind everybody, Murano was the study which looked at the relapsed refractory patients, and generally they're patients who've not seen BTKI, because in fact it was done in parts where there wasn't really access to ibrutinib. So these were patients who generally had mainly only chemoimmunotherapy, and they then either went on to get BR as the standard relapse therapy, which that's what we gave. You did not give FCR twice, so you gave BR, and this was compared to venetoclax rituximab, 26 cycles of venetoclax and 6 doses of rituximab. And just to look, but actually this is now being presented, and there's some very impressive progression-free survival curves here. The blue curves, 55 months is the median progression-free survival, but virtually everybody is relapsing, interestingly. That curve does not really look like it's got a plateau. There are a few patients who do seem to not relapsed again, but what this is showing, I mean, I guess it's these patients who initially got chemoimmunotherapy, is that this regime is certainly not curing them, but it's holding their disease really nicely, and this is the overall survival curve. In a way, what's more relevant is, when do you start retreating patients? Well, I don't know if it's more relevant, but on this regime, it's a median of 63 months before a patient goes on to yet another line of therapy. And I'm just going to touch on what we might be able to deliver when I've done these slides. And MRD showing it's a really good predictor for response again. So those patients who don't obtain MRD negativity, or what's defined as MRD high in this study, so that's greater than 10 to the minus 4, not actually that many patients, but they progress very rapidly, whereas those patients who are undetectable MRD, and these will be blood studies at the they're doing much better. And there were a few patients with sustained UMRD, but they're the usual characters. They're the patients who have got mutated IGHV, or they wild type TP53, and they're just, in general, they're not those patients. As I said earlier, we're serving them very well with our current treatments. There was some retreatment data presented, and in the study, some patients could go on and get VR if they relapsed, and although they responded, they all relapsed again relatively quickly. But showing it's feasible to give VNR again, and some of those patients did nicely. But I think in reality, in the relapsed area, one would normally then go and switch to an alternative therapy. And this was from the Seymour publication in Blood last year, showing that in fact all of these patients who hadn't seen BTKI before were responding nicely. So very quickly, just a couple of thoughts on the relapsed patients that we see in the clinic here today before we just go on to some new agents. There are patients who will have received chemoimmunotherapy. They're still very much around. Patients who are relapsing on ibrutinib or ACAL, or intolerant, of course, of the BTKI. And I think there was a question about that in the chat. And I think if you're intolerant of one BTKI, it's very reasonable to then reach for an alternative. And I think we're soon going to have 3 available, ibrutinib, ACAL, and zanubrutinib. So I think we need to be thinking about that. Frankly, there are not very many patients that we've yet seen in the UK have relapsed after VO. And really, this slide, which I actually slightly modified just before I showed, because I realized the previous version I had was rather old, but we've got some really good long-term follow-up data now on both BTKIs and now on VR, and they have got different qualities. You don't get MRD negativity with BTKI, but does that matter? You do with VR, but they still seem to relapse. We now know a lot about the kind of things patients will get in terms of side effect profile, and there's loads of data out there on what we can give afterwards, and that's a lot of real world data which many people in the UK community have been very influential in producing. But there is a small group of patients who I'm going to call the ultra high risk. We used to define those in a variety of ways. How do you define them? To me, they're the patients who are, and this should really say covalent BTKI, BCL2 inhibitor, and anti-CD20. And these patients do not do well. Patients who have seen those agents and are progressing, they do progress, and we need to find other ways of managing these patients. And I just put that up here, that we know now quite a lot about clonal dynamics of patients and what happens genomically to them at the various stages of treatment. But you get a different type of mutation resistance pattern depending on what you've given. And this was a nice article by Francesc Bosch and Ricardo Dalla Favra, showing that the mutations or the pathways that get affected by the different treatments that we deliver and what you see. And this slightly complicated slide shows that we know now that you get mutations both in the BTK, particularly C481S for ibrutinib and acalabrutinib, and other either bypass pathway activation kind of activations or mutations in PLC gamma 2, and similarly there are mutations within BCL2 that confer resistance to venetoclax. So what options have we got? And we're getting increasing data on the non-covalent BTKIs. There's data coming out on BTK degraders. And we have got immune-mediated therapies, which in CLL, although I will argue they started there, actually have been not as far developed as we're seeing in the remainder of the lymphoma field, or indeed myeloma or ALL. And that CAR-T, although it kicked off in CLL, has been a laggard for CLL, although there's some interesting data coming out suggesting it might have a role. So what are non-covalent BTKIs? And it turns out that they actually bind and inhibit the kinase activity through a completely different mechanism to the covalent ones. So the covalent ones are ibrutinib, acalabrutinib, and zanubrutinib. And it appears, and this was data which was presented at EHA on the back of this publication, that pirtobrutinib, which is a non-covalent BTKI, interferes with the folding of the BTK protein and inhibits its kinase activity in quite a different way. And that's probably why it works in patients who are resistant to the covalent BTKIs, because the mutation pattern is different. But it's not that you do not see mutations on non-covalent BTKIs, and in fact, you see a much wider range of different mutations with the non-covalent BTK, and it probably is something about that mode of action that results in that. So whereas in ibrutinib and ACAL treated patients, you really see the C481S mutation. You do not see those same mutations with the non-covalent. And it'd be interesting if we bring the non-covalent further up in the treatment pathway, what we're going to see. And this was data from Jennifer Brown, which showed, this was at EHA, that showed that the C481S clones would disappear with treatment with pirtobrutinib, but you would get new clones growing out. That's actually the C481, which was going, but all these new mutations coming. And so there's going to be a whole industry out there looking at these mutation patterns. Very briefly on bispecific and trispecific antibodies, lots of excitement of that in diffuse large B-cell lymphoma and in follicular, and I think remarkably little data on their usage in CLL, the concept being that you can bring a CD3 cell and a tumor cell together with your bispecific. There is some data on epcoritamab in Richter's, and I'm sure that Nick may discuss that tomorrow. And in fact, this is the study which is looking at that, and it was also used in CLL. And essentially, this study showed that it was tolerable, and it said encouraging antigen. And then there's this efficacy in Richter's syndrome potentially. But I bring this up because, in fact, one of our concepts for a relapse refractory study in the UK is to combine pirtobrutinib, so I talked about that, with venetoclax, and then see if we can use epcoritamab as an MRD consolidation, to try and obtain MRD consolidation in patients who do not obtain MRD negativity on this combination. I think I have got a few more minutes. I might watch this 12:03. Says we've been presenting for 39 minutes, but let me just quickly finish off these last slides. There's no way this morning I can go through all the things that could be considered for treating CLL, and I think I've really tried to focus on things that we've been doing in the UK, and also trying to put that in the concept of today's clinical practice, that these are just some of the pathways internally that one might be looking at. I think the BTK degraders are something we're going to be seeing coming soon. And these, again, have a completely different mechanism of action against BTK, which is an essential pathway downstream of the B-cell receptor, which is what drives a CLL cell and keeps a CLL cell alive. So interfering in that pathway is such a potent thing in CLL. And essentially, this will result in degradation of the BTK molecule as opposed to inhibition of the kinase. And there's a company called Nurix, but you will find that all the other companies are following up with their own BTK degraders. So it's going to be the new pathway on the block for us to look at. But there was some data presented showing how BTK activity is degraded, and there's some very initial preliminary data in multiply resistant treated patients responding to that, which was presented at ASH last year, and I'm sure will be presented again. I'm going to very quickly finish on CAR-T, and that is because there is some latest data out on that. And I've said this before, this was the original excitement where CAR-T suddenly seemed to work people, and it was in CLL patients that we first saw that happening. And this is from the original New England Journal publication now in 2011, showing resolution of the bone marrow and reduction of that. And just to say that it's been updated last year, that some of those patients who received the original CAR-T infusion are still in remission, no evidence of CLL, and interestingly have circulating CAR T cells. Turns out they're perhaps not what one might have expected. Turns out they're cytotoxic CD4 cells. But I think this just shows that in some patients, this is clearly a highly effective treatment. I think sadly, one of these patients may have died due to a COVID infection, which shows that immunotherapies can have other issues. But I just wanted to share with you the Transcend 004 data, which was presented at ASCO and ICML recently, and also recently published in The Lancet. And this is using Lysocell, which is a mix of a CD4 and CD8 infusion, manufactured from CLL. So this is autologous CAR T cells, no other agents like ibrutinib. There are other studies around where ibrutinib, for instance, has been added, patients given a fairly standard regime. And just to show that, again, in this study, there are some patients who are doing extremely well in terms of PFS and overall survival. But these are the patients who get the, they get a complete response, and they are unfortunately the minority of patients. And this also correlates with their MRD levels. But those patients who don't get a good response or don't obtain MRD negativity with CAR T are sadly progressing quickly. And I think, again, this is encouraging data, but we need to really work out why are these patients responding so well and these ones so badly. And there's a lot of work to be done there. There clearly is quite a lot of work going on, because I was amazed that there are 74 trials in CAR T and CLL active at the moment. So it's available and around in a trial setting. And people are increasingly looking at how we might be able to change that. I think I've had my 40 minutes. So it's a bit of a skate around in different areas. And I hope I've covered everything that we did. But, clinical trials have been absolutely pivotal in the development of rational CLL therapies, underpinned by understanding of biology. I am a little concerned that academic studies are more and more difficult to deliver, and are moving away from the other studies, which are very much led for other reasons. And we want to do studies which we are then able to transform that into therapies for patients. So I think we need to think very carefully about this as a community. As a more general point, and I think we've already raised this, our patients do really well with, most of them do really well with the therapies we've got, but there are a small group of patients out there where we're going to show, and I just tried to give you a little bit of a flavour of where things may be going. Very finally, there are combinations of different BTKIs with venetoclax out there, but the data is still very immature, and I think we'll learn about that more and more over the next year. Thank you.
Fantastic overview. That is really, really useful, particularly as a springboard for us now to talk a little bit about where we are going with CLL. We've lost Helen because she had to disappear off with a family-related pre-planned event. So, Piers, I'm just going to immediately just put you on the spot. I very much do like the way you said the problem we have within academic, and of course you're hinting at the pharmaceutical industry is required really to run trials that meet licensing stipulated guidelines. I mean, and quality metrics. That's just the way it is, and it is forever a problem. We're seeing that across the board in the lymphoproliferative disorders. If a trials group runs a trial that might get quite an important finding, I think you said Hodgkin lymphoma with the German BRICAD study, it's a long way from showing a large, big randomized trial showing benefit for patients and then actually being able to use that approach in your clinic. That's obviously a disconnect that none of us are going to be able to solve at individual level, but it is something we do have to keep reflecting on. And I think in CLL, an important example, which you're hinting at with the static trial, is an everyday practical thing. We don't really know if you're on a BTK and you've been on it for 3-4 years and you're particularly first line, these are not the unstable relapsed refractory, but the first time BTK patient, do they actually benefit 10 years from now? Are they better off keeping on at 3 years or stopping? And that's why I thought it's a good start point before we go into the more complex, because actually static is asking a question the pharmaceutical industry really should have addressed a long time ago. I mean, the Elevate trial should clearly have had an amendment to randomize all patients still on drug at 3 years to stop and restart or carry on. So what are your overall comments on that?
Well, I think one issue is, I mean, it's interesting when you look at some of these studies that you think if you do an early readout, you think that things are equivalent, and then you do a late readout and it's changed. And a good example of that, I think, is the Elevate Tumor Naive study, where we thought that obinutuzumab had no additional role and now we're seeing those curves divide. Now, I know they're not yet significantly different and probably never will get there because of the numbers, but from a hypothesis generating thing, you suddenly see that. And actually, George, I remember you telling us all we should be giving obinutuzumab to our ACAL patients at an ASH, and everyone looking at you going, why would you do that?
I could obviously read the future, Dr. Patten.
And so I think long-term readout is really important, because if we read out too early, we might get the wrong message about study. So, and I think that's going to be particularly tricky actually with something like static, because my gut feeling from my own practice is, and I'm just going to use Flare as the example. I have a number of I haven't got static open. I will open it shortly. I therefore have a number of patients who've stopped ibrutinib at the end of 6 months, and by and large they've done absolutely fine. And in fact, nobody who stopped it for coming at the end has required retreatment. I do have a patient who has progressed quite clearly and will need treatment soon, but everyone else, and in fact some of them have even gone from being MRD positive to MRD negative, but there may be other reasons for that.
6 years to close. You said months as just a miss.
Yeah, sorry, 6 years. But the problem is, of course, that's just in the first year and they're all doing fine. But in fact, I don't know if in 5 years' time that's going to have been the right thing for them or not. And that's why I still think we need to do that study.
But just to bring this point, of course, there are other healthcare environments around the world with co-pays and things. There are a lot of reasons why people stop BTK. And, interestingly, I guess I feel bad using examples from the private clinic where we've had first-line access to BTK a bit longer. I've now got a collection of patients who have decided at that 2-3 year point to simply stop. And, small anecdotes, but we commonly see these patients not relapsing and remaining well. So it's a very practical. So I think static is a really good practical, but it's going to need long follow-up. So I'm going to bring Ros in on this talk. So I think, Ros, you and Helen both showed the long-term follow-up of CLL14. And I think it speaks to Piers' point about this long-term review. I think it is quite interesting. I've always had this view, and I think speaking to what Piers was saying, that I was a bit more of a fan early on about the obinutuzumab acalabrutinib, because this perception that when CLL patients go through a cycle of relapse, they become more unstable in many ways, whether it's infections, autoimmune, hospital trips, the lymphadenopathy, etc. And presumably the only explanation for the drop-off of the curves with CLL14, so we track them and then the overall survivals are clearly separating. That must be an excess mortality of those chlorambucil obinutuzumab patients as they're cycling through a series of relapse. Do you have any views on that, Rosalind? I mean, you showed us that data.
Yes. I mean, it's interesting, isn't it? You're postulating that controlling the disease better promotes genomic stability and therefore prolonged survival.
Yeah, it's more this question, because it's always.
I mean, this is the whole conundrum, isn't it? Because why do BTK inhibitors work for genomically unstable disease? Why is continuous BTK therapy, where you have detectable levels of disease that should be evolving, why does it appear to be better? I mean, there must be things that we don't understand yet, because you can apply that logic to the CLL14 curves, but it doesn't make any sense if you try and apply that in the BTK situation. So I think it's fascinating. I think there's still, we still clearly haven't got to the bottom of what's really important. We've picked up a few things that clearly are important, but there are still things that we clearly don't understand. We're measuring some things, but maybe we're not measuring all the right things quite yet.
Yes, and I think I guess one of the points that I dwell on is just the general medical aspects of relapsing patients. I think that's if you're on your continuous BTK and you're utterly fine, you're 75, you're taking your tablets, and then we come along and say, okay, we're going to stop them or put you in a trial and randomize stuff. You can imagine some patients are going to say, you know what, I'm all right. I'm all right. Why do I want to stop these tablets? And I guess I'm speaking to my uncertainty that we know that might increase their risk of early relapse and then going through that whole phase of relapse and being ill and et cetera, et cetera. But
I think the other thing to say about relapsing patients now is that because we're not getting people that have been through FCR and BR and had all the kind of infectious complications and issues with that. I think hopefully our relapsing population is going to be a fitter population going forward than it was kind of 10 years ago. So I think that's good for patients as well, isn't it? It's, they're coming into those subsequent therapies hopefully with less infectious insults and things over time than they might have had on the older treatments. I don't know.
No. Good points. Right, Nick, I'm going to bring you in before I bring Lilian in. I mean, Teague said there's all this interest in BTK. Pharma loves BTK. Biotech loves BTK. I guess it's because we've shown time and time again, the more refined you are at targeting BTK, the fewer off-target effects you have. And I guess toxicity kills an early phase drug, a biotech drug, a lot quicker than lack of efficacy. So we're being driven from the biotech industry up to keep looking at BTK. And I know Nick has got a lot of experience with pirtobrutinib. We've shared some patients have gone in your trials. We're all getting experience with these newer BTK targeting drugs. What's your thoughts? What do you think about these non-covalent BTKs, Nick, where they're going to fit in the algorithm? Because obviously Piers is talking about some quite dynamic trial designs, but a bit closer to 2023. Where do you see them coming in?
Well, obviously they're going to come later, following BTK inhibitors by the way the trials have been conducted. It's an interesting thing to consider whether or not they will completely displace BTK inhibitors at some point or not. It's a very interesting point to consider. I have no answer to that yet, and you don't know if you're going to have a BTK sort of covalent BTK inhibitors that would be active if people progressed on non-covalent BTKs. So I mean, watch the space. I mean, for the moment, I think I think the BTKs are safe. Covalent BTKs are safe for a few years, but I think that these are going to come into the frontline study. And more importantly, the degraders. I think the degraders are a glimpse of the future that are going to be there all over the place, and they can manage to engineer the degrader to target any molecule they want, really. So we can potentially see it against Notch, against BCL2, against any other molecule you want. So that would be very interesting to see where that goes.
Yeah, I think it is a fascinating space. I might bring Piers back in on because obviously perhaps the non-covalent BTKs that are a bit behind the curve, like nemtabrutinib, they're obviously pushing to try and jump up. I think one of their combo trials they're hoping to get open later this year is nemtabrutinib with venetoclax against venetoclax rituximab. So trying to sort of squeeze space, and you fully understand why they're going to do that. It's interesting, I think, and pirtobrutinib, they've obviously been quite brave in the mantle cell space, haven't they? They've gone head to head with ibrutinib Acala in a big global phase 3 randomization, because I'm just not sure biologically. We all see the nice pharma and development slides telling us that a non-covalent, why it might be better than covalent, but I guess none of us really know, and I'm slightly uncertain. But it's a sort of brave trial. Go on, Piers, what do you think about?
Well, I mean, I think it's fascinating, because it's a new kid on the block, and the Germans are going to use Perto frontline in their next study, in the CLL18 study. But it's looking at that mutation pathway, the paper in New England from Wang and the presentation at EHA. This is a whole host of array of new mutations. It doesn't seem to have a pattern and stuff like that. Whereas actually with the covalent BTKIs, very clear. You get your it's basically your C481S there, which is causing resistance in CLL, and we know a lot about that. And I'm just wondering that we may jump ship to the non-covalent, knowing much less about them, and actually turn out to be not quite what we expected. So I mean, I guess I'm just being a little cautious about
on the BTK mutations. Appears there's been that data set suggesting ZANU might throw up a different set of mutations.
Yes, it does seem to. I mean, I think you still see the C481S, but you also see a couple of other ones as well. And that may mean fundamentally it's working slightly differently. But whereas ACAL and ibrutinib seem to be very similar in their mutation profiles, and ZANU is not. So I guess it's got that different one as well. And in fact, there is a concern, I think, that the ZANU may have caused mutations which we know that the non-COVID, the PERTO may be resistant to.
Yeah, so in these conversations and worry we're going to be like CML doctors and obsessing about sequencing BTK. Yeah, you can see a whole new world arising. Leila, let me bring you in. So you have been a fantastic trials patient. You saw some of your trial data being presented there from Clarity. What are your views from a patient perspective hearing all this? Does it inspire you? Does it concern you?
Absolutely, it's great to hear. Before I retired, I was a clinical trialist, so just to declare my colours. So I'm with Piers. I passionately believe in trials as a key step in the way forward. I think we should have pragmatic trials that are as closely, certainly the late phase studies should be as closely linked to the real world as possible. I think statics are hugely important trial from the patient perspective, so it was good to hear that. And one of the things that first made me stop and think and feel it was important was issues of resistance. Having been 51 when I was diagnosed, I'm interested in the long term, and it also brings back to one of the things that was mentioned at the beginning today but hasn't really been picked up, I think, which is the sequencing of treatments. So I think there's, I would echo the caution about taking what's, to a patient perspective, relatively short-term follow-up. I remember when you hear someone saying we've got good follow-up, it's 23 months, 30 months, even 60, to be honest. It doesn't really feel very long from the patient perspective. And can I just throw in a bit of a googly at the end, which is progression-free survival is a really difficult outcome for patients. I have, after 15 years of having CLL, 30 years of working in trials, I don't really understand what it means for me as a patient.
So Leila, let me actually really nail that down, because I think this is also a naughty side of the pharmaceutical industry. Well, actually, that's not fair. Actually, I'm going to say the regulators, because progression-free survival, as was first introduced to us with CLL4, as I love those slides, Piers, that is brilliant. And also the comment that they started off with overall survival as an endpoint. You didn't mean it in a derogatory way, but of course, it was the endpoint.
Yeah,
actually,
pretty final.
That's pretty final. And when you say to patients, they do say, well, is this going to make me live longer? That is actually an endpoint that patients are very interested in. But
also, it doesn't tell you anything about quality of life. And that's what's key if you've got CLL, which was most of what we've talked about today, to be honest. And progression-free survival tells us nothing. I don't want to just...
But my other slightly cynical comment on progression-free survival is, since the regulators have accepted PFS as an endpoint, it's meant every registrational trial is giving us these 3 monthly CT scans. And it's why you get that completely artifactual drop-off of the glomerular obinutuzumab curve and GLOW, because that is your end of treatment. You get your CT scan, and 40% have got a lymph node that is increased by X percentage.
No, absolutely. And actually, the PFS in the German studies is not CT driven.
And in the PFS, of course, they did get 12 months of glomerular in the CLL14 trial, but you see, you have a much smoother curve. So that's driven
because it's clinical progression and not CT driven progression. We're talking about two completely different things.
And the other point that those of us with long enough remember is to remember when obinutuzumab was being pushed with chlorambucil, and Roche loved that data to remind us that progression on a registration scan could still be 18 months before treatment. And time to next treatment is a lovely endpoint, but it's very soft and it's very hard for regulators to work with because it's so variable as to when people actually, when the clinician, when the patient, et cetera, decides they're going to retreat. And also, slight dig, the Germans are really good at time to next treatment because they include death or time to next treatment. Pharmaceutical industry loves to remove death, so you'll have a trial where you can have a time to next treatment with 95% at 3 years, but actually 7% or 8% of the trial have actually died, but they're not in a time to next treatment because they haven't had more treatment. And when Ottman presents his data, you'll notice from the CLL, he always starts by saying time to next treatment includes patients who have died, are included as a time to next treatment event. So, oh dear, it's Ros.
I think that's one of the difficulties with what I always say to my patients as well. The difficulties with trial data in general is it's helpful for us to discuss what the right thing to do is, but actually for an individual, for you as a patient and as an individual, you'll only ever be one dot on that curve. So what happens to you, and I think trial information in general is it's difficult, isn't it, how to apply that to you as a person rather than
binary. What happens to you, isn't it?
Make general recommendations.
And I think that's this whole thing about sort of prognostic data sets to predictive at an individual level, and it's why I guess the appeal of things like MRD, etc., because that you go from big, broad data sets to an individual patient. And if you have had treatment X and you've cleared the disease from your marrow, and you as an individual know that you have done well. So you sort of then can hone in on what's closer to what's happening with your dot. But the big challenge is when you're at the beginning of that curve. So you're a patient with your clinician trying to make a decision. What you do, exactly what you said. You are a dot out of 353 dots on the trial. Where are you going to sit? You know, you could be one of those amazing patients who 10 years from now, brackets, if we have the follow-up, brackets, are still actually in ongoing remission. But ever so challenging. Now we are getting very close to time. Piers, can I just check on other combinations? So the other BTK inhibitors, venetoclax, do you have any trial development comments, or we're just going to have to wait for the licensing trial?
So yeah, I think we will. I mean, the one I didn't talk about here at all because we're not exploring it, and is for triplets. So IVO, and I think we've got a lot to learn about that because so far the German data and actually now the Alliance data is saying that isn't showing any benefit over a doublet in terms of IV or VNO, and there's toxicity with that. So the patients you're going to, so I would have been a great believer in triplets a few years ago and would have said this is what we need, but actually just shows you can get that wrong. Now, what we don't know is, because a lot of that's to do with toxicity, are we going to then get a sort of AVO combination and stuff, which may be quite different. And I think we will learn about that relatively soon. So we'll see different BTKIs being paired up with venetoclax, and indeed different BTKIs paired up with different BCL2 inhibitors. BeiGene are very keen on that. And as a UK group, we will be closely involved with them on developing that pathway. But because it's still tentative, it's difficult to say too much about.
I'm very rudely cutting you short because as a chair, I like to stop on time. So what a fantastic morning, Piers. That was an amazingly useful walkthrough. I think being able to talk through all of these points we've discussed today with experts from across the UK, and also having Lelia representing a patient voice in such a clear and educated way is tremendously useful. There's only one question left in the chat, which was about MBL. How often do we monitor them? Is it the white cell you monitor? So there is split views. MBL, I really try to demedicalize them, and I personally try not to keep them in the clinics. Others might have different views. I know MBL patients have a slightly higher chance of chest infections, hospitalization, if you follow them over a long period of time. So that's sorry, we have to round off. So out of time on that one. My final comment, apart from thanking everyone on the panel, thanking the CLL Support Association, Lewis behind the scenes has done all the technical things to make this work. Please, can people put feedback in? So this is only the second year we've run this meeting. The whole idea is it's meant to be really approachable, accessible. People, when we can talk about whatever we want to talk about in the world of CLL, we're not restricted. And so we want that open format to continue. Do you like the breaks? Do they work? Would you prefer more intense? Would you prefer less talk, more chat? More talk, less chat. We've gone for the balance at the moment that we think is right, but it might not be right. So do please give us some feedback. And on that note, thank you to the participants for putting in the questions. Thank you to the CLL Sport Association, and thank you to my panel members.